So for those of you that don't know me, I'm Sarah Cook and I'm an advanced pharmacist practitioner, and I work cross-site. And I'm going to deliver the talk that I intended on delivering a couple of weeks ago about dosing in renal replacement therapy. So hopefully I can give you an overview of the renal replacement therapy modalities that are out there, demystify the simplicity, it's not very simple, and talk about the variables affecting drug clearance for RRT. So we'll go through the modalities, drug considerations, patient considerations, and circuitry modifications that you might notice in practice, and touch upon some new developments, which will probably be a whole different talk in themselves. So we'll just touch the surface with some of those. So the intermittent modalities that are out there, intermittent hemodialysis, peritoneal dialysis, and SLED, and the continuous modalities that are used are SCUF, CVVH, CVVHDF, and CVVHD. So at UHL, we use a few of those only. So we narrowed our choice a while back to CVVHDF. Not many, I don't think we've used CVVH in many a year due to simplifying the options for nursing staff and to avoid confusion. But within CVVHDF, we use citrate as the anticoagulant predominantly across the trust, and heparin anticoagulation for patients with significantly impaired liver function or ECMO patients. And only about 20% of drugs studied in CVVHDF actually have any evidence-based dosing info acquired so far. Intermittent modalities that are new to the Glenfield site currently, and new guidances in the making, include intermittent hemodialysis and SLED, and Ricky Bell is leading on that project at the Royal, at the Glenfield as we read, and we have been discussing about increasing the training across the sites to enable that to happen. But the intermittent modalities, patients don't tend to tolerate the big fluid shift, the big hemodynamic shifts, the sicker they are. So it would be suitable for a smaller number of patients. So the trial that they will undertake at the Glenfield will inform our choice of patients accordingly. And within the SLED kind of cohort, only about 1% of drugs are studied for the appropriate dosing. So that's going to be really quite difficult to dose patients very accurately. So I would imagine that the majority of our sick patients would remain on CVHDF, but we'll see how that goes. So a nice little recap. This is an amended picture. I didn't do it myself. It's from the Tawani paper. I've just adapted the slides slightly in purple so you can see where I've annotated them. So hemofiltration is a convection-based system, and hemodialysis is a diffusion-based system. So in a convection-based system, you rely on a transmembrane pressure gradient. So you've got solutes moving from an area of their high pressure to low pressure across the semi-permeable membrane. Whereas in diffusion, it's a concentration gradient. So a high concentration to a low concentration. And in a diffusion-based system, you've got a dialysate to provide that concentration gradient, which is running countercurrent to the patient's blood. So you can see in hemofiltration, a convection-based system, you're going to remove much larger molecules because you've got solute drag across that pressure gradient. Whereas in a diffusion-based system, you're looking at your smaller molecules that you're removed. Now, I've kind of plotted the options that we would technically have at UHL, although we don't, as I said, we don't really use the CVVH as such, but we do use the CVVHDF. If you look at the CVVHDF, we're using both convection and diffusion. So that's why it's really useful in an intensive care environment, because you're getting the solute removal via convection, and you're getting the water removed, so you can offload patients as well. And you're looking at those larger molecules as well. So yeah, at the top you've got your high transmembrane pressure, your pressure gradient, and your concentration gradient with diffusion. So can we just titrate to effect in ITU? That's what we do a lot of the time. So for things that are that we've got a decent, reliable pharmacodynamic effect that we can measure. So blood pressure, heart rate, heparin assays, APTT, blood glucose, we can do that, yes, for sure. But we particularly find with renal replacement therapies, it tends to be the antibiotics and antifungals that tend to cause a bigger problem for us. All of the things that we monitor for infection, we've got delayed measures. So we'll have a lag with inflammatory mediators, and to get MCNS from the lab, PCTs, beta-D glucans, and even to some TDM as well sent away, it can be really, really difficult. So we find, definitely as a pharmacy team, we find that we get involved with CRRT with respect to antibiotic and antifungal dosing. And then, of course, toxins. We don't always filter patients because of renal impairment. They might need filtering to remove toxins. So we'd need to understand the PK/PD of the drugs and how it interacts with the filter there. So, as I just mentioned, so what's the indication for the renal replacement therapy? Is it an AKI? Is it just fluid removal, toxin removal, or an acidosis? Sometimes post-surgery, certainly I've seen at the Glenful some of the cardiacs that come out, you don't necessarily have a really significant AKI, but they might be profoundly acidotic and they'll have a spin on a filter. That kind of tends to help, but not necessarily have a great impact on their AKI. They not necessarily have a huge AKI. So it kind of really depends on what you're aiming for, really, for filtration. So your patient might have a decent amount of intrinsic renal function. So do we use creatinine clearance in urine output? I would avoid creatinine clearance. Often that's old. There's obviously a lag with that. Urine output is usually a good test of whether their kidneys are actually working. But urine output also can be related to the amount of fluid that they're driving that patient. So that can be problematic itself. And then you've got the issue of augmented renal clearance. So if you're filtering a patient that does have some background intrinsic renal function, you're actually giving them much better renal clearance than they actually have at baseline. So you might end up with much higher clearance of drugs than a normal healthy patient. And that tends to be with your younger patients. I often see that with sort of hyperdynamic patients as well. So going on to the drug PKs, there's so many variables, a little bit of a whistle-stop tour now. So volume of distribution. So the lower your volume of distribution, the greater your effect of RRT. And that relates to your lipophilicity, so your log P value that you can look for. So a high log P means that the drugs are lipophilic, more lipophilic. So tend to kind of, it will be a positive log P rather than negative log P. Negative log P will be Lipophilic, you're probably looking at anything with a log P of about sort of 2-ish to really kind of impact greatly. So the higher the volume of distribution, the lipophilic drugs will have a higher volume of distribution versus hydrophilic drugs. And those lipophilic drugs will be found intracellularly and in adipose tissue. So they're not being spun through the filter. So there's lots of drug elsewhere in the body. The clearance route, if it's renally cleared, of course, the filter sets are made to mimic the glomerulus. And the way that the filters are set up, if you think about your pressure difference between your afferent and your efferent arterioles and your glomerulus, that to maintain that pressure gradient, it's all to mimic the kidney. So if it's renally cleared, yes, it's likely to be removed by RRT. Then protein binding. So this is only really significant when you look at drugs that are about sort of like 90% protein bound. You do tend to get some removal. It's only the free fraction that's available to be cleared. So high protein binding, RRT, for RRT is less important. And then your molecular size, which was on the other slide, so versus your pore size and your modality. So when you look at like vancomycin in intermittent hemodialysis, it's got known pore clearance, not pore clearance, as in pore, pore clearance due to its molecular weight. However, when you start to look at it on a CRRT circuit, where you've got, especially with CVV-HDF, as we discovered when we introduced CVV-HDF, we were going from like being really, really cautious on even on CVVH with vancomycin to like just wondering where the hell this vancomycin was going. So it really does demonstrate quite well the difference in molecular weight. This is the really, really boring stuff that you don't really want you don't be calculating at a bedside, but actually you can use the Siffing coefficient to work out how much is being cleared, and the Siffing coefficient it basically gives you capacity to it's an indication of the capacity of the drug to pass through the filter. It's a saturation coefficient rather in hemodialysis, and it's obviously PK dependent. So you can do these fancy calculations. You can go and look on your filter. You can have a look at what your ultrafiltrate flow is, and you can do TDM with your ultrafiltrate versus your plasma, and you can work out your Siffing coefficient for the drug, and thereby work out your clearance, which is great. And I think we should be doing that more often. And that's where a lot of the PK/PD studies, go down the route of lots of TDM, obviously. But we just don't have access to, timely TDM. So in the future, as Hussein Muller would kind of go down that road, it would be pharmacogenomic testing of patients at the bedside and bedside assays, which would be great. So we look at sickness severity. Our patients are often heavily fluid resuscitated, so that, needless to say, has a higher impact with don't know what's happening there. That's a higher impact with hydrophilic drugs because you've got a greater dilution there. Not entirely sure where. Sorry, techno problems. So that's where the idea of front-loaded dosing regimes comes from, and additional loading doses. So you'll probably often hear, and I know it's in my kind of line is go big or go home in the first 24, 48 hours. Don't compromise on your dosing, just get it up there. go in with normal dosing. We'll talk about fluconazole in a moment. You have to go in with 50% higher dosing in our patients, but we can end up in a pickle, you know, where when you look at the pharmacokinetics of a drug, you can end up staying below, for instance, the MIC for like the majority of the dosing interval, if we're not, you know, trying to dose appropriately in those first 24, 48 hours. You'll never achieve it. So I think what I'm trying to say is be brave, especially in the first 24, 48 hours, and then we can consider where we are with the patient and decide whether or not we want to back off. By that stage, you know, an AKI might have resolved, it might all be sepsis driven, or things might have declined even further, but we kind of really don't know in those kind of first 24, 48 hours. So I generally say, let's go for it. So then the anomalies then, so I don't know if anyone here has had the joy of dosing fluconazole on a filter or being tapped on the shoulder by a pharmacist and asked to increase the dose, and you kind of think, are they absolutely bonkers? But it's basically because it usually, without the filter attached to the patient, you've got tubular reabsorption, so you've got a nice long half-life there. So you need 50% higher doses on the filter. So talking about antibiotics and PD indices, so I think in Kevin and Piyush's talk, they talked about you know, time dependent and concentration dependent kinetics and drugs that have a mixture. So as I was mentioning about antibiotics, it's really important to get above the MIC and get this indices right, whichever it fits into. So, you know, for vancomycin, you need to stay above the MIC for the whole of the dosage interval, whereas things like aminoglycosides, you need a nice big whack of a dose and get above that kill dose level to stun the bacteria, and that the effects will last for the dosage interval. You don't necessarily have to stay at the high concentration, and then you've got the anomalies that have a mixed picture. So it's useful to know some of those bits and bobs, and you can compare the MIC that the lab measure versus the UCAS breakpoints. So then looking at how other variables for filtration and how doses are how drugs are handled in RRT, looking at the filter surface area is something that you don't usually get involved in, but certainly during the pandemic, when we had shortages of the ST150 sets, we had to switch to the M100 sets. We just basically had to take the sets that were available to us. So if there is such a problem as that, you're being advised that on the filter paperwork that you're having to like use a different set. Bear in mind your drop clearance might be affected. So if it's the number ST150, M100 is a lower surface area, so you're going to max out on that filter sooner. So you're going to end up removing less drug. And then your duration. So typically CRRT is supposed to be continuous, but we all know that we get filter downtime. We get filter downtime with you know, going to CT, going to theatre. If patients are profoundly you know, hypercoagulable, or if they're you know, they're profoundly inflammatory, for instance, the COVID patients, the filters clog and they clot, and then we get downtime. And if the nursing staff can't save the filter, obviously then we've got the delays in setting up another filter. So that can actually affect your clearance as well. Now moving forward with the project at the Glenfield, with intermittent hemodialysis and SLED, it's going to be interesting with how we monitor and dose there. And that is a huge piece of work that we need to do as a team. But it's going to be ideal for patients that can rehab. So we won't necessarily have to keep them on CRRT like forever and ever, or waste filter sets for CRRT, or filtration fluids for CRRT. We can spin them on an IHD or SLED circuit, but we will have to be cautious with those patients that we select. So dosing, the KDIGO guidance recommends that you go for a 20 to 25 ml per kilo per hour effluent volume for CRRT. Now, some of our regimes do have the option for 35, a lot of them do, and actually from a sustainability perspective, we probably should be looking at, you know, aiming for the 25, but looking to make sure that the patients have the right style, the right modality of renal replacement therapy. So if they don't need continuous, why do we need to be using, you know, 35 ml per kilo per hour on them like continuously for 24 hours a day, when we could possibly spin them through IHD cycle or SLED, and use far less plastic, far less fluid, and impact our wastewater systems less as well, and therefore possibly have less of an effect with drug clearance as well. So we can time doses of drugs that are affected by IHD and SLED to after the spin on the filter, rather than during the spin on the filter, or some drugs will need additional loading doses after. So I think we've covered downtime, haven't we? So I'm not sure if you've ever got involved in this pre-post dilution choice. I think a lot of the protocols are actually specified, and you just sign your life away on the documentation. But if you've got your replacement fluid post dilution, it actually gives greater clearance. And I think that's the way that we're set up. So it's like a rule of like 2/3. So I think we've got 2/3 post replacement, post dilution, and 1/3 pre dilution. And that works on the principle that you've got more blood, more concentrated blood versus your dialysate. Can't speak today, sorry. And then there's your effluent flow rate and your dialysate flow rate, obviously, therefore, as well, impact your drug clearance. There's some really good documented evidence for that. However, it's worth noting that with CBD-HDF, the clearance is not additive. So just because you've got convection and diffusion, it doesn't mean you have 100% of each effect. Your regime is tailored accordingly to the proportion of each. So looking at new technologies then, I'm not sure if anyone has been involved in any of these bits and bobs. So Oxiris filters are filter sets are predominantly sort of geared towards your septic patients that are really, really hyper-inflammatory. And the Glenfield ECMO team tend to use these. Now, what we fell down on was going for the 72-hour expiry for your filter set, but actually not realising that for maximal effect for these filters, the filter sets, you need to change them 24-hourly. Otherwise, they are designed to pull all of those inflammatory mediators off, which coats the Oxiris filter and clogs it. So you will end up with downtime. So you need to anticipate that downtime if you're using an Oxiris filter, and make sure your nurse at the bedside knows to change it 24-hourly. So if you get asked to prescribe an Oxiris filter, it's always worth prompting and making sure you document, you know, that we need to change that filter set next day. Once the patient becomes less, you know, less hypercoagulable and less, you know, hyperinflammatory, you can just leave it running for your 72 hours, but you really just do need to anticipate that, especially in the first few days, if you're using Oxiris for a septic hyperinflammatory patient, that it needs a 24-hour change. they're expensive, but patient selection is key. And I think we've got a nice little algorithm for those filter sets if you ever get involved in those. The cytosorb sets that you can actually rig into the RRT filter circuitry. They're really, really interesting, and I'm going to actually put it on the website that I've put together because there's so many drugs that are absorbed into these cytosorb filters. I think like they've clearly been working on it, because I think last year it was such a small number of drugs that they had evidence for, but now it's building and building and building, and it really is worth keeping a close eye on that. That is definitely something that we need to be cautious of going forward. And specifically, I think the initial reason why cytosorb was kind of rolled out was to pull off things like apixaban and your DOACs and stuff for aortic, cardiac surgery, basically. So that's where we've been kind of using them in the trust until now, but definitely state of the art. They were really pushing it for lots of other indications, and I think that's probably going to be something that we see a lot more of. So I'll keep the website up to date with that, because if there's something that is really, really important for your patient, then we need to kind of think about other drugs that aren't. There's no evidence for absorption, but just because there's no evidence for absorption, it's probably that we've not tested it yet. And then TPE. And this is where I said at the beginning, there's probably a whole that's probably a whole nother talk in itself. But often for RRT, it's worth noting that it's particularly for patients that have like vasculitis and stuff, they're often on renal replacement therapy. Then they'll have a spin on Plex, which is the other name for TPE, and then, even though we said before that the protein binding wasn't necessarily significant with renal replacement therapy, if you've got a patient that's having intermittent TPE or Plex in between, anything that's protein bound will be pulled off with the albumin. So it will impact. And then of course there's ECMO, which is again definitely a whole different ball game and discussion. You've got so much extra circuit there that you'd absorb to. So I think it's easy to say that it's a very rubbish film. So I think you probably wanted to come here today and kind of know how to simplify your kind of approach to renal replacement dosing. Arsha pharmacist. So that brings me on to a little bit of PR. So I've put together the ITAP's website. I'll show you it in a bit. So on there is a dosing adjustments page, and our current list of antibiotics for our CRRT dose adjustment table is on there, and it's antibiotics basically. Now, as we said, with the project at the Glenfield for IHD and SLED, and also with TPE in mind, and I know that is something that we're keen to roll out across both the ITU sites. We need to look at an all-encompassing document really that you can just refer to, and choose your filter modality, choose your filter modality and make your adjustments with antibiotics. And similarly, TPE, a lot of the issues are with antibiotics as well, antimicrobials. So that is something that I'm going to pop up on the website. So you don't need us when we're in bed at night, and you can find the answers yourselves. In most cases, it is going to be antibiotics that are really, really pressing. Other things probably can wait until we do a little bit of research the next day. I've also I'm pursuing a subscription for a an online reference source called Critical Illness. And I've actually been involved in writing some monographs for the team there, and it's all specifically for critical illness dosing. So you'll find all of these pharmacokinetic parameters within the reference source. And also what is great about it is that it will give you the information not just in healthy subjects but in the critically ill population. And I think I mean I've popped the SPC website, so medicines.org.uk, and the MHRA website, but it's worth noting that a lot of that information is like, you know, population-based kinetics in healthy subjects. Some of them will allude to the fact that, you know, in certain disease states the PK/PD might be different, but they're not as useful as something like critical illness. So hopefully we'll get that. And I've repeated that. That's probably impressive. Yeah. So that brings me to the end. Does anyone have any questions at all?
I just have a question about you mentioned about there's like an intermittent one that we can use that is just almost as effective.
Yeah.
But then when would we choose to use that? Because we don't really seem to use that much.
Yeah, so the project at the Glenfield that Ricky's working on at the moment is, so you've got your intermittent hemodialysis, so you have big fluid shifts, so a lot of particularly ill patients don't tend to tolerate those fluid shifts. But the SLED is the slow, low efficiency daily dialysis, and you can actually pair that up with some diafiltration as well. And need to clarify before we do all the dosing guidance whether Ricky intends to have the diafiltration or just the dialysis. But yeah, in those patients, so if you've got patients that you need, you know, to offload, the ideal, you know, if you've not got any, you know, significant worries about your hemodynamic shifts, basically, that's where the beauty of that lies, and patients that you want to rehab, really. It's ideal. The filters, as you know, they're noisy, they're horrible, you know, to just, you could either choose to rest someone overnight or spin them overnight or spin them for short periods of time. When you're looking at SLED, it's like 8 to 12 hours of therapy. So you could time that 8 hours, have your rehab, you know, yeah. And then, you know, it depends on what the issues are with the patients, whether it's rehab, whether it's sleep. You know, if patients are delirious, you might want to prioritise sleep and not, you know, have a filter pinging off next to their head all night. So I think it's going to be really interesting to see how that goes. But again, it's the selection is key, and it'd be interesting, I think, to see what we can do with it. But I am concerned about the drug dosing element, because there is such a low, there's only like 1% of drugs have been studied in SLED, whereas IHD, you've got a bit more information. It's so much more similar to, you know, standard HD 3 times a week that your renal patients will get. So there's a lot more info out there. But yeah.
That's why I'm saying
because the filters we use you change it very lot, very expensive.
Yeah.
If you can just have a system that you just do for a short period of time.
Well, it doesn't actually need to be finished.
Yeah.
And it is reducing how much you. There are other reasons why she's not rehabbing, but well, we don't have access to that other.
Not yet. But I think.
Is it the same machine?
No, so it's a different machine. So we purchased the machines probably about 2 years ago now, I think. And due to one reason or another, it's just not, you know, it's just not got. They've been serviced, and we've got the plumbing sorted now. They use fewer fluids, fewer consumables. You're not spinning them for so long. But I think we got about 12 machines. So I mean, not up to me, but you know, sharing is caring. I think it would be ideal really to move. Yeah, I don't know.
Yeah, there is plans. Yeah, we keep working.
So is that the right person to do that?
A lot of people tied up into those. We need the right structure.
Yeah, definitely.
The water supply will be. Yeah, we had the road that's almost comes in. We had, yes, it's quite off the sinks
Yeah.
Yeah.
in here and in recovery.
Yeah.
So it may come at some point. I mean, it's not that.
No, it's not. But it would be good for like with
It's like weaning for it.
yeah, I get another one. I want to go to pick my own
I think
tree.
we've got one or two.
Mm.
It's a handful a year.
Yeah, I mean, definitely those types of patients are, you know, the ones that we really need to kind of champion having those kinds of modes. Especially, you know, how many times do we end up like just spinning patients like past their baseline, like creatinine, and you know, what are we aiming for with some of these patients? We can back off and, you know, either filter them less frequently or use, you know, SLED or IHD. So yeah.
Thank you.
Yeah, definitely. TDM and bedside. Yeah, I think pharmacogenomics is going to really kind of, you know, take off bedside tests. So Hussein Mullah is a clinical research pharmacist that's based at Glenfield, and he's doing he's liaising with industry to try and get a bedside vancomycin point of care testing, and look at also the you know the genomic CYP cytochrome P450 genotype monitoring, and looking at the combination of bedside testing and genotyping to kind of stratify what kind of doses that we actually you know, aim for with patients. So I think that's quite an interesting development. There's going to be lots of that, I think, in the future, but how well invested it will be from industry for, you know, certain drugs that come on the market, or how it will be pushed from the NHS and what capacity we have for that will be